This is the prokaryotic cell structure. Prokaryotic cells are about 10 times smaller than eukaryotic cells. A typical E. coli cell is about 1 micrometer wide and 2 to 3 micrometer long. Structurally, prokaryotes are very simple cells when compared with eukaryotic cells, and yet they are able to perform necessary life processes. So, the reproduction of prokaryotic cells is binary fission. Binary fission is a simple division of one cell into two cells after DNA replication, and the formation of a separating membrane and cell wall. All bacteria are prokaryotes, as are the archaea.
Let's go through the parts of the prokaryotic cell. So first we have the flagella. So the flagella is referred to as organelles of locomotion or cell movement. It is a thread-like protein appendages that enables bacteria to move. So this is the flagella. Next is the pili and fimbriae. So the fimbriae is a hair-like structures most often observed in gram-negative bacteria. They are much thinner than flagella, and it enables bacteria to adhere surfaces. Next, the sex pilus. Here. So, sex pilus is a single structure that facilitates transfer of genetic material from one bacterial cell, or the donor cell, to another, or the recipient cell. Next is inclusion. So inclusion is the storage of excess nutrients. And then the plasmid here. So the plasmid is a small circular molecule of double-stranded DNA. It is referred to as extra chromosomal DNA because it is not part of the chromosome. Next, so the capsule. So glycocalyx is a thick layer of material located outside their cell wall. It is slimy, gelatinous material produced by the cell membrane and secreted outside the cell wall. So there are two types of glycocalyx, namely the slime layer and the capsule. So, slime layer. The slime layer is neither highly organized nor firmly attached to the cell wall. It easily detaches from the cell wall and drifts away. It also enables certain bacteria to glide or slide along solid surfaces and seem to protect bacteria from antibiotics and desiccation. So, capsule. Capsule is the knowledge of the chemical composition of capsules. It is useful in differentiating among different types of bacteria within a particular species. So, for example, H. influenza. It is a cause of meningitis and ear infections in children, and are identified by their capsular types. A vaccine called Hib vaccine is available for protection against disease caused by H. influenza capsular type B. So it can be detected using capsule staining procedure, which is a type of negative stain. So the other parts of the prokaryotic cells were already discussed on the first video, which is the eukaryotic cell structure. So prokaryotic cell. The prokaryotic cell is the simplest and most ancient cells. So it is smaller than eukaryotic cells and had simpler structure. It lacks or no nucleus, and it is a single circular chromosome. So the inside of the prokaryotic cell is basically an open unit with no compartments. There are no membrane-bound organelles. However, small structures called ribosomes are scattered throughout the cytoplasm. The cell's DNA is located in the region of the cytoplasm called the nucleoid region. The nucleoid region is not the same thing as the nucleus because it's not enclosed by a membrane. Prokaryotes also have a cell wall that surrounds the plasma membrane. The composition of the cell wall depends on whether the cell is an archaean or bacteria, but basically it consists of complex polysaccharides. Prokaryotes also often possess one or more flagella, which are used for movement.